Hi everyone, in this video I will be going through the structured questions for the RE on genetic inheritance. Here is one of the first times you encountered something called a pedigree. A pedigree is essentially a family tree, showing you the history, the records of who has particular disease, who is healthy in a family line. The reason why pedigrees are so useful is because it helps us predict the future and also helps us trace the genotypes and phenotypes of various family members. We cannot perform test cross, as you can probably imagine, when it comes to human beings. So we cannot quite derive and figure out a human's genotype through a test cross. However, we can find out through pedigrees how the history affects the present lineup of children. So in this example, we see a particular disease called the Tay-Sachs disease. It is a neurological disorder. And the first question, in fact, already asks you an important question. Is this disease allele dominant or recessive? Explain your answer using information from figure 1. All right. So when you look at pedigrees, you often find squares and circles. They will always give you a legend, so you can figure out even if you forget. Squares represent male, circles represent female. If you ever see shaded, means that is the affected individual, whatever the disease may be. Okay, in this scenario, it's the Tay-Sachs disease. Okay, so is this disease causing allele dominant or recessive? Okay, let's reason this through, okay, to solve this puzzle. Now, let's say this disease is dominant. Okay, I'm going to write big H as dominant, small h as recessive. So let's say this disease is dominant. If the disease is dominant, okay, that means you just need one copy for it to show, right? So that means to say all the affected individuals will be either heterozygotes or homozygous dominant, right? Only two possibilities. If the disease allele is dominant. Okay, one more individual here. However, that also implies that for an individual to be normal and healthy, their genotype would be definitely a homozygous recessive, right? Every healthy individual needs to be homozygous recessive. Now, does this make sense if we look at this current pedigree? Let's say an allele is dominant, right? Something doesn't quite play out over here. We look at this set of parents and children, for example. If the disease-causing allele is truly dominant, right, then two normal parents that are homozygous recessive cannot possibly have an offspring that has a disease, right? Because both homozygous recessive, inevitably the child would definitely be homozygous recessive too. Yet this is not what we see. So therefore, the opposite must be true. It must be true that the disease-causing allele is recessive. Okay, so how are we going to explain that the disease-causing allele is definitely recessive? Right. Usually, the best way to explain why a disease-causing allele is recessive is to search for carrier parents. What do I mean by carrier parents? Okay, carrier parents are parents that are heterozygotes, right? And that means to say that they contain the disease-causing allele, but on the surface, their phenotype is normal. Okay, this is what we mean by carriers. So the best way often to prove that something is recessive is to search for such parents. They are carriers. Both parents are carriers. If you look at 7 and 8, both parents are carriers, right? And then there's a child that has the disease. So that means this child is definitely homozygous recessive, right? In fact, you can label all of them already. Right, so for this child to be homozygous recessive, it must have received one copy from mom and another copy from dad. Yet, we see that their phenotypes are normal. So that means this allele is recessive, right? Because even though it's present, we see that the phenotype is normal. Okay, so if I show you a sample of how you can answer this. Okay, number one, the disease causing allele is recessive. So here's a suggested answer. There are other ways you could answer this, right? As long as it justifies what you've claimed. For example, we say for individual 12 to be affected, he must have inherited one copy of the disease causing allele from individual 7 and 8. Since 7 and 8 must carry one copy of the allele, and yet the phenotype shows that they are normal, that means to say the normal allele is dominant, right? And the allele that is disease causing is recessive. Remember, when we go back to the pigeon game, right, we always define, right, an allele that's dominant is the one that shows its phenotype in a heterozygote. And the recessive one is the one that's phenotype is masked. And this is exactly what we see. Even though one of the alleles are present, we do not see its phenotype on the surface. That means it must be a recessive allele. Okay, so number fourth point you could try to add also. Consider adding. If the allele were dominant, at least one parent of individual 12 must be affected. Which makes sense, right? If it were dominant, right, at least one of the parents would have to be affected. Yes, yet that's not what we see. Okay, so this is one suggested answer that you can give. State the probability that individual 6 is homozygous for the hexa gene. Explain your answer. So where's individual 6? This is individual 6. Okay. Here we say that the probability is 0. How's that so? Okay, if the mom is homozygous recessive, right, because she has the disease, we see that one of the child is homozygous recessive. So that means dad definitely has one copy of the recessive allele. To have children that show the dominant phenotype, that means dad also has one of the dominant alleles. So this is the genotype of dad. So what's the probability that individual 6 is homozygous? Actually, a very, very low chance. 0 chance, actually. Because for individual 6 to show the dominant phenotype, has to inherit one of the big H from dad, and definitely will inherit one of the small h from mom. Okay, so the individual is likely 100% a heterozygote. So you see, given individual 2 is affected, which is mom, her genotype must be TT. In this case, I use HH. Okay, whatever genotype you want to use. Therefore, individual 6 must inherit one of the alleles from individual 2. Since individual 6 is normal, her genotype must be heterozygous. Or for me, I put big H and small h. Okay, finally, let's look at C. What is the probability that the next child born to 7 and 8 is a girl who is affected by Tay-Sachs disease? So, individual 7 and 8. This individual 7 and 8, we see that their genotypes are heterozygous in nature. So let's put the two parents together. Okay, if we look at the Punnett Square, these are the possible genotypes of the children. Right? There is a 1/4 chance that the child would have Tay-Sachs disease. And we are asking for a girl, so there's a 50% chance that the child will be a boy or a girl. And therefore, the probability here is 1/8, which is 0.125 probability that the child will be a girl and have the disease. The remaining questions are all questions that require you to draw genetic diagrams, which you have had the chance of practicing. In this particular example, it's about a cross between a homozygous bull with red coat and a homozygous cow with white coat. And the offspring are all a mixture of red and white hairs, which is what we call roan. We've went through this example in class. This is an example of co-dominance because both red and white traits were equally shown on and expressed in the phenotype. Using appropriate symbols for the alleles, draw a genetic diagram to illustrate a cross between a roan bull and a white cow. Okay, a roan bull would have to have both forms of the alleles present. A white cow was likely homozygous for the white allele. Notice here, when we define the alleles, we use a big C, and then as a superscript, we use R and W. The reason why we use the same alphabet as big C for both instances is because neither of them exert dominance over each other. Just like in the example that we went through in class. Okay, so when there's no complete dominance, then you would have to use such a form of symbol. Okay, so let's just do a quick check. You need to have all your titles at the side. Okay, and then you need to make sure you have all the crosses to show that it's mating. Make sure you use the right symbols. Okay, make sure also that you have lines to show that the gametes are forming. Make sure that you circle your gametes. Make sure you draw the lines to show the crosses that can possibly happen. Write down all the possible genotypes. Write down all the possible phenotypes. And then write down the expected ratio. Okay, in this case, it's a 1:1. Alright. So you would also want to write something like that. Roan coat is to white coat. So we know what the ratio represents. Okay, so this is one example of drawing genetic diagrams. Next question also requires you to draw a genetic diagram. A male and a female with normal color vision each have a father who is colorblind. Okay, so this is a colorblind example. So remember here, when we say colorblind, the example we taught you, this is sex-linked. More specifically, is linked to the X chromosome. Okay, and it is a recessive allele. So you need an individual that is like that for females to be colorblind, or in the case of males, a genotype like that for the individual to be colorblind. So show the possible phenotypes and genotypes of male and female children from these two parents. Okay, so what is the nature of the genotypes of these two individuals? First and foremost, we see that both of the dads of these two parents have the disease. The color blindness. So that means the genotype will have to be this. Recessive and also with one Y chromosome. Okay, what about mom? Okay, mom has normal color vision. So we don't know at the moment what mom may have. Okay, but first and foremost, we know that for males to be males, he definitely needs to inherit one Y chromosome from the father. Okay, so this Y chromosome definitely went to this guy over here. What about the X chromosome? What X chromosome did he inherit from the mom? The X chromosome that's inherited will have to be a dominant allele because the individual is not colorblind. Okay, so it doesn't quite matter whether we know or not whether the mom is a heterozygote or homozygote for the normal vision. Okay, what about the female? The female will likely have inherited one copy of this from the dad Okay, there's no other way about it. Couldn't have inherited the Y chromosome. So what did she inherit from the mom? Well, she definitely had to inherit a dominant version of the allele from the mom, right? So even though we don't know what's the genotype of the mom, it doesn't matter. What matters is that we know that this individual is normal, so definitely she has at least one copy of the normal allele on the X chromosome. Okay, so these are the genotypes of the parents. You're supposed to then draw a genetic diagram based off it, right? So here is the one for the dad, here's the one for the mom. Okay, again, you need to have all the headers at the side. Make sure you include all the X to show the mating. Make sure you have all these arrows to show the gametes forming and have circles surrounding the gametes. And also have all these arrows, sorry, not arrows, these lines to show the possible combinations that can occur. After that, you want to write down all the genotypes, all the phenotypes, and then after that, write down the expected ratio. Okay, now moving to the last question. Okay, this last question is on blood type. We've encountered blood types before, so let's draw a genetic diagram for it. Explain what is meant by the terms gene and allele. We've defined this before. So a gene is a sequence of nucleotides which make up a stretch of DNA which codes for a polypeptide, whereas alleles are alternative forms of the same gene. So using symbols, draw a genetic diagram to show how two parents may have children with all four blood groups. The only possibility is if you have a parent who is heterozygous and with the phenotype of blood group A and blood group B. Then you can have all possible blood groups. Okay, this is not a difficult genetic diagram to draw. Make sure you look out for all the standard features that you need to find in a genetic diagram. And you should be able to get this ratio of phenotypes. Okay, you can pause the video to be able to look at the answers in depth. But I'm going to move on to the very last part. Describe how a single base deletion in the allele IO leads to synthesis of a non-functional protein. So a single base deletion, right, what will happen is that, let's say this is the gene, okay, just one segment of the gene. And if we have a single base deletion, right, one of the bases get deleted, everything that we read now will be of a different frame. Right now we're reading a different frame. So definitely what you cause is a frame shift. Same thing if you have an insertion, you will also get a frame shift. And it can be quite drastic because when it's a frame shift, you change the sequence of the codons. And as a result, the sequence of amino acids that will be coded for. And as a result, you can change the structure of the shape of the polypeptide chain, and which may render it non-functional. Because at the end of the day, you must remember that DNA sequences will transcribe to form mRNA, and then mRNA will be translated to form polypeptides. Okay, so that's all for going through RE 7.1. If you have any questions, feel free to contact me. Thank you.